focus to our free demo class. Today we will speak about the genetic code, which is our 7.1 in our text, in Kaplan's text. So we have some learning objectives I want you to know. Okay, in these learning objectives we identify what we want to do in this lecture. Okay, so what are our learning objectives? Our learning objectives that you have to differentiate between three types of RNA, which are the mRNA, the tRNA, and the rRNA. You have to transcribe a DNA sequence like GAAT-TCG into its mRNA conjugate. Define the concepts of wobble and degeneracy. Identify the translation outcomes of key codons including AUG, UAG, UAA, and UGA. Predict the likely impact of different mutation types on the resulting peptide. First thing, we have something in science called the central dogma of the cell. Okay, focus with me on this, please. Central dogma of cell. For the central dogma of the cell, there is this analogy. Okay, what is this analogy? That if we imagine that your DNA is your cookbook, it is the book that contains every and each specific variation, each and every specific feature of you. Okay, this cookbook, if we want to imagine your mother is going to make you pasta, for example, she has the cookbook. The cookbook has the pastas, it has the cakes, but your mom doesn't do all of the recipes inside the cookbook. It only makes the recipes that she inherited from her grandmother or she know from her parents, whatever. Okay, so the cookbook is all the stuff genes. Okay, all the stuff genes. When we make DNA and we make it to RNA, this is called transcription. So we say that it is transcribed. Transcribed into what? Transcribed into the pasta paper. The pasta paper. So your mom will open the cookbook, it will go to the pasta paper, which we call the genes, and this is an active gene because it contains information, and she will look for the method to make pasta. And then when it makes the pasta, which is the RNA, into protein, which is the pasta, okay, let's imagine this is pastas. Okay, so we said this is translation, that the code has been translated. This is the central dogma of the cell, which we identify an academic way like this. We say that we have three processes happen, and these processes are DNA can replicate itself. When does this happen? I am building the foundations of science with you now, so I want you to focus with me. Okay, this is a rapid review. The replication of the DNA happens before cell division. Okay, why? Because we have to preserve the number. So for example, if this cell wants to make a copy, we call the complete set 2n, make a 2n and the 2n cell, which is called mitosis. You have to go through DNA replication. If we want to make DNA to RNA, it's called transcription, and this is very important, transcription. If we want to make RNA to DNA, it's called reverse transcription, which is found in some viruses, which are RNA, and then they go back to DNA inside the host cell, which makes them very difficult to attack. If we make RNA into protein, it's called translation. It's called translation, and this is the central dogma of molecular biology. Now, if we see a DNA molecule, okay. You will find that the DNA molecule is a double helix structure. We have two strands. Okay, a strand from 5 determines the phosphate, 3 determines the OH. Here 3 is the OH, of course, and here 5 is the phosphate. Okay, 5 is the phosphate. So what type, or what is, because DNA is double stranded, right? DNA is double stranded. RNA is single stranded. This is basics of science. So what is the strand that we will use to make RNA? Because we have double strand right here, so we must use one strand only to make the RNA. This strand in any prototypical gene, what we call a prototypical gene, okay, is that RNA is synthesized in the 5 to 3 direction, and it is anti-parallel to the 3 to 5, which we call the template strand on the DNA. So when we have two DNA strands like this, one from 5 to 3 and the other is from 3 to 5, the one that we actually make the RNA from is this one, because it will make a 5 to 3 RNA. And this one, this red one, we call template strand, because it is the template. It is the template for the RNA. The 5 to 3, we have two terminus for any polypeptide. Okay, so any protein, polypeptide means protein. Okay, polypeptide means protein. So any polypeptide has two ends. One is called the N terminus, the nitrogen terminus, and one is called the amino terminus. Okay, and one is called the carboxy terminus. So C. So the polypeptide chain is synthesized from the N to C terminus, or from the 5 to 3 direction. So let's summarize this in a figure. Shows our figure 7.2. In our figure 7.2, okay, this is our figure. So DNA, which is in the 5 to 3 direction, is called the coding strand. Why it's called coding strand? Because if you imagine, this strand will be the same as this strand exactly. So this coding strand, which we call coding, will be identical to the mRNA except T for U, because RNA doesn't have T. So in DNA we have T, but in RNA T, which is thymine. In RNA we have U, which is uracil. Okay. I wish I was seeing you, because I know in the live session you have the ability to ask questions. So this free demo is not what I like to do, but it is necessary to comprehend you with the knowledge to assess how the tutor is in this course. So this strand will be identical to this strand, except in so we call it coding. Why it's coding? Because the mRNA codes for protein. The mRNA is the code, so we call it coding protein. But where do we make the template? What is the template that we make from? It's the 3 to 5. So we call it the template strand. It makes the template which is anti-parallel and complementary to the mRNA. So the DNA template strand is complementary and anti-parallel to the mRNA. So T is A is T, no, it's U, C is G, C is G, and you know the game. A binds with T and with double bonds, and C bonds with G with triple hydrogen bond. And instead of T in the RNA, there is U, okay, in the RNA, so you don't have to conflict that, okay? So for each A you find the DNA, you have to replace it with U on the DNA, okay? And then when we translate that code, so did you understand what I just said? When we translate that code, we get the protein, so it's called translation. Okay, and this is the direction of transcription. It's from 5 to 3, and from direction of translation is from the N terminus to the C terminus as well. Okay, so this is the genetic overview. Okay, now let's go to another topic, which is the types of RNA. The types of RNA. There are 3 types of RNA in the cell. There is the messenger RNA, there is the transfer RNA, and there is the ribosomal RNA or RNA. The messenger RNA is called the messenger because it carries a message. Okay, so messenger RNA, it carries a specific message. Okay, it carries the message from the inside the nucleus. As you know, the DNA is inside the nucleus, inside the cell. So after the transcription of mRNA and of course post-transcription modification, it goes out through some nuclear pores into the cytoplasm, right? Into the cytoplasm where it is translated. So the message is carried from the nucleus into the cytoplasm. This is the cytoplasm, this is the nucleus. So that's why it's called messenger RNA. Okay. It is transcribed from a template DNA using something called RNA polymerase. So the enzyme is, let's go for the different color for the enzyme. So the enzyme is RNA polymerase. Okay, RNA polymerase. Like the replication, when we take replication, or it was explained in previous chapters, it was done using DNA polymerase. So for replication we use DNA polymerase, for transcription we use RNA polymerase, and hence the name, it synthesizes RNA, so it's called RNA polymerase. Okay, of course, if you want to transcribe any DNA to mRNA, any tRNA to mRNA, okay, so any DNA to mRNA, let's copy this image. So you have the double strand, you have the DNA 3 to 5 and 5 to 3. What is the template that you will use? You will use the 3 to 5, of course, as we said before. Okay, to make the 5 to 3. So the A, it will be T, no, it will be U, T, A, G, C, A, U, G, C, T, A, C, G. As we said, A with T, C with G, and then instead of T in RNA we use U. This is the whole game. Okay, using RNA polymerase, of course. Okay, RNA polymerase, and the how this works is every 3 letters are called codon. So this is codon number 1. This is codon number 2, and so on and so forth. So each codon, okay, each codon can give more than one amino acid. Okay, this is the idea I want you to grasp from this. Each codon can give more than one amino acids. They are represented by more than 20 codons, and I will explain the idea behind this, but it's not on your level. So each codon can represent like one amino acid can be represented by more than one codon in eukaryotes. Okay, in eukaryotes. The mRNA is monocistronic. What does this mean? It means each mRNA as a whole gives one protein. So we have an mRNA, for example, insulin. We have one for cholesterol. We have one for progesterone, which is the pregnancy hormone for females. We have, for each hormone or enzyme, we have an mRNA that codes for it. Okay, in prokaryotes, in the bacteria, in the fungi, okay, in bacteria. So in bacteria, in prokaryotes, okay, can be polycistronic. What does this mean? It can be translated at multiple times in the same time and generating different proteins. Okay. So what about tRNA, transfer RNA? This one is the one carrying the amino acid to the ribosomes to make the amino acids. Okay. This tRNA has an arm. This arm has something called anticodon, and this anticodon is charged or activated when it is connected to the connected to the codon of the mRNA. Okay, and it has another arm to bring the amino acid. Okay, so let's see how this is done. So this is the image of the tRNA. This is the structure, and as you can see, this is the structure of the tRNA. We have from the 5 to 3 end. At the 3 end we have the OH, which is where the activated amino acid is attached, and at this point we say that the tRNA is charged or activated. Okay, we have an anticodon sequence pairs with codons in mRNA. This one is what pairs with the mRNA itself when it is on the ribosome. So imagine this is the mRNA, 5 to 3 direction. There will be a sequence on the mRNA which is GUA. This GUA sequence will attach with this sequence, and the amino acid that was activated on here will be dropped on the mRNA like this. Okay. What is the enzyme responsible for charging the amino acid onto the tRNA? It's called focus with me on this enzyme please. It's called the amino acyl tRNA synthetase. You have to know the name for this enzyme, so let's give it a color. So amino acyl tRNA synthetase, it is the enzyme which is responsible for charging the tRNA with amino acid for proteins. The final one for protein synthesis, the final one is the ribosomal rRNA. We have the ribosomal RNA, which is the rRNA. Okay, in the ribosomal RNA, we know that this is, if this is the cell, we know that this is the cell membrane. This will be the nucleus, and inside the nucleus there is the DNA, of course, and the chromatin, and we have the nucleolus. Okay, we have a structure called nucleolus. Inside the nucleolus, the ribosomal RNA, the ribosomes are made. Okay, in the nucleolus. So the location is the location of synthesis, I mean, is the nucleolus. Okay, nucleolus. And the ribosomes is a complex machinery. Okay, it's a complex machinery to assembly the proteins, to assembly the codons. Okay, so it to assembly the proteins from the codons. I mean, so they have a very strict structure. I want to describe it now, but it's not included in this free demo. The structure of the large subunit and the small subunit and the mRNA between them. So this is explained more in our course. The course that comes in a very discounted price at $4.99. We have 12 weeks of live sessions, of exams, of mocks, of 1000 AMT style questions, everything. Okay, so in this free demo, I'm just explaining that it's a complex machinery to form a peptide. They act as something called ribozymes. Okay, ribozymes. So they are made of blocks of RNA instead of peptides, and they help in the make of the peptide bonds between amino acids. So they help in the making of peptide bonds between amino acids, and they splice out the introns. Introns are the areas of the gene that doesn't code for protein. So we splice them out in the post-transcription modifications, and we will take the post-transcription modifications of mRNA. This so this is the thank you for your listening to this chapter, to this small introductory video about our course. Of course, the PDF you will be found it's below, and you will find the quiz after the chapter. The quiz will be open to the people who enrolled in the course. So I hope to see you in the next one, and I hope you understood the genetic code as an essential molecular biology concept to understand everything about genes, DNA, how things are inherited, inheritance, everything. Okay, thank you, and see you in the next one.